Hello and welcome to our next section in our evolution unit. We're going to discuss today the fossil record and geologic time. We're going to probably begin here with a review of prokaryotic and eukaryotic organisms and cells. Remember what those were. And we're going to end our section with hopefully understanding radiometric dating and how it is we come up with the ages of organisms and their first appearance in the fossil record. So this picture right here, let's just start right here. This is a picture of an incredibly old fossil. This fossil was discovered very recently in 1993 in Western Australia, and it challenged the current thinking because it was older than anything that had been discovered to date. And it is determined to be a prokaryote, some type of prokaryotic organism. And it was dated at 3.5 billion years old. So that is older than what had been thought of as the oldest life at the time. So currently, we're looking at prokaryotic organisms first appearing on Earth sometime around 3.5 billion years ago. This structure was found in what are called stromatolites. And you may or may not have heard this word before. Stromatolites are dome-like structures. They are, here's a picture of stromatolites. They're produced by bacteria that basically deposit minerals that they collect from the seawater, and it forms these little dome shapes here. These stromatolites we see being formed today by bacteria in Australia as well. They're thought to be, there are also places that fossils have been found in Utah and in Canada, these similar types of fossils. They're thought to be prokaryotic. And just to quickly review, remember when we talked about prokaryotes and eukaryotes, and we're comparing these two different kinds of organisms. Prokaryotes are a lot smaller, like 1/10 the size of eukaryotic organisms. Prokaryotes are more simple. They have no membrane-bound organelles. They do not have a true nucleus. They just have a nucleoid region where their genetic material is. Eukaryotes have a nucleus. They have membrane-bound organelles. Likely, the prokaryotes in this very, very, very old fossil would not even be as complex as modern prokaryotes. It's thought that perhaps their genetic material isn't even located in a nucleoid region. It might just be randomly scattered throughout the cell. So prokaryotes, if you remember, bacteria, like when we talked about the 5-kingdom classification system, Eubacteria, Archaebacteria, or as in the 4-kingdom classification system, the Monera, those are all prokaryotic organisms. So quick review for you. If you remember the scientist by the name of Carl Woese, we talked about him when we were discussing classification systems, and he did a lot of work studying prokaryotic organisms. And he suggests that the first organisms may have been methanogens. They are prokaryotic or anaerobic bacteria. They obtain their energy by using carbon dioxide to oxidize hydrogen, and they, in that process, produce methane gas and release that methane into the environment. Some of these, we see these organisms today, and remember our early environment, early Earth environment doesn't have oxygen. So perhaps these would have been some of the first organisms. They certainly did not need oxygen to thrive. Today we see some of these near hydrothermal volcanic vents where the temperature of the water is far in excess of the boiling point of water. It's very hot. So methanogens could have survived very hot conditions as well in that early Earth atmosphere. So that is one idea of some of the early life that may have existed. Here we have just a picture of methanogens. Here is a picture of what they would look like under an electron microscope. Very tiny, little prokaryotic organisms that we see today and could have been some of the first forms of life on Earth. So when we look at our geologic time scale, and I do not expect you to memorize this at all, so don't worry, please don't copy all this down. This is a lot of information, and a lot of it is just FYI for your information. These are the main eras that we're looking at in Earth history. These are some of the events that happened in those eras. They're broken down into periods and epochs or ages. We're not going into that at all, but I want you to at least have a broad understanding of the kind of time that is being discussed here. When we look at this first prokaryotes appearing at 3.5 billion years ago, it's roughly about here. Here's the formation of Earth, 4.6 billion or 4,600 million would be the same thing. Make sure you can do those conversions. We talked about those earlier in the year. First eukaryotes would not have come around until we see those in the fossil record about just a little over 2 billion years ago. So that would be right about there. We're going to do a little project where we make a geological timeline to give you an idea of how long ago this is compared to some recent events and what that looks like. So we'll be working on that in class, but I do not expect you to memorize all of the information on this chart. One big question I have for you, how are these timelines determined? And remember I talked about radiometric dating? Well, we're going to look at that right now. Radioactive isotopes. You've heard of these words. What does that mean? What is an isotope? Remember from the very beginning of the year when we talked about atoms and different kinds of atoms. If we have one kind of atom, say a carbon atom, and we look at another carbon atom, they have the same number of protons, but sometimes they have a different mass. That is an isotope. The same type of atom with a different mass. The reason they have a different mass is that they have a different number of neutrons. Those different masses sometimes are radioactive, meaning that as they decay, they produce radioactivity. Carbon-14 is one such radioactive isotope. It is a carbon atom that weighs 14 atomic mass units, and when it decays, it produces nitrogen and a beta particle, which is the same thing as an electron. And you don't need to know that specifically, you just need to know that it produces something that's radioactive. So you probably have studied this before. Half-life of a radioactive isotope means the time it takes for half of the radioactive matter to decay, meaning the half-life of this atom would be the amount of time that it takes for, if we had a big lump of carbon-14, a big pile of it, half of that would have turned into nitrogen-14 and radioactive particles during its half-life, during the time of its half-life. The half-life of carbon-14 is 5,730 years. So here's a little problem for you. See if you can figure this out. If the amount of carbon-14 in a sample has decayed to a level that is 25% of what the original sample would contain, how old is the sample? Now, here's a nice little graph that you could use to answer that question. See if you can figure that out on your own, but I will tell you. So if you want to pause it and think of the answer. If you use this graph, here we have the percent of carbon-14. At 100%, you have your full pile of carbon-14. After this many years elapsed, after 5,730 years elapsed, you should have 50% of that left. 50% remaining would be the nitrogen 14, its product, but you would only have 50% left. So, as the question said, what if your sample has decayed to a point of 25%? How old would it be? Well, it would be 11,460 years old. You would have gone through 2 half-lives to get to that amount of the original. If a specimen is 17,000 years old, approximately, how many half-lives would have elapsed? Well, if we look on the graph, we have a roughly 17,000, just a little over that right here. We'd be right here. We would have gone through 1 half-life, 2 half-lives, 3 half-lives have elapsed to get to that age. So how do we use this information to determine the age of fossils or organic material that used to be a living organism? How do we use that? Here's how it works. A very, very small fraction of the carbon dioxide in our atmosphere contains some carbon-14. And it's formed because cosmic rays collide with the nitrogen atoms, and we get this heavy carbon-14 isotope. So when plants and other autotrophs absorb this radioactive carbon-14, they incorporate it into their tissues. So animals and heterotrophs absorb it by consuming the autotrophs. And so all living organisms have a certain amount of carbon-14, this isotope, in their tissues. When that organism dies, that carbon-14 that is in it, already in the organism, it does not continue to gain carbon-14 because it is dead and it is not carrying out any kind of metabolism. Instead, the carbon-14 decays at a known rate. Remember, 5,730 years for a half-life. That is the known rate of decay. So, when the amount of carbon-14 is measured in the remains of an organism, that can be compared to the amount that you'd expect there to be, and you can determine the age of that organism. Now, you've probably realized that, and actually the graph, if you even go back one slide there and look at that graph, obviously carbon-14 has a useful range, after which it is no longer useful and it won't help us to determine the age. And this is about the useful range, 100 to 30,000 years. After that, we really can't use carbon-14 to help us. We need to use some different radioactive isotopes. And especially since we've been talking about things like, billions of years, we need something that has a much longer half-life to be useful. There are a number of isotopes that are used: potassium-40, rubidium-87, and uranium-238 and uranium-235 are used for longer ranges, for more, because their half-lives are considerably longer. So when we measure, the way these work, uranium is a really good, useful radioactive isotope for determining really old rocks. When we measure a sample, what we can do is measure the amount of uranium, for example, 238, that is present, and we can compare that to the amount of lead 206 as well. And by comparing the amount of daughter isotope that is formed against the amount of uranium, the original parent radioactive isotope that is there, we can indicate the age of a rock, and therefore also help us to determine the age of the organisms that are, or the relative age of the organisms that are in the rock as well. Some advanced ideas. These are some things we didn't talk about, but they go along with everything in this section. What are endosymbionts? What is the endosymbiont hypothesis or the endosymbiont theory? You can run across it called either one. And why is that significant to the process of evolution? What do people think may have happened there? And that has to do with eukaryotic organisms. research what else is produced in addition to the daughter isotopes in some of the radioactive decay of these heavier radioactive isotopes. Research any interesting fossils that have been discovered and find out how scientists determine the ages of those fossils. And any other good questions that you have. And I look forward to seeing you in class, and get ready to work on our little timeline activity and make your own geologic timeline.